Hello everyone. In population genetics, the Hardy-Weinberg principle, also known as the Hardy-Weinberg equilibrium, states that the allele and genotype frequency in a population will remain constant from generation to generation in absence of other evolutionary forces. We'll briefly discuss what allele is and what genotype is. But before that, let us know about the history of Hardy-Weinberg principle. This principle is named after the G.H. Hardy and William William Weinberg, who first demonstrated it mathematically. So let's understand what is an allele. An allele is an alternative form of the DNA sequence on the located sequence of the same gene located in the same gene locus on the homologous chromosome. So these are the homologous chromosomes, and the allele is the alternative DNA sequences which you can see located in the same gene locus. So this is capital S, small s. So these are two different types of the DNA sequences. This is one form of the DNA and this is its alternative form of the DNA. This is called alleles. In the diploid, each individual carries two alleles per gene, forming AA or AA. If two alleles are same, they are known as homozygous, and they are different. If two alleles are same, as we can see here, they are homozygous alleles. If they are different, they are known as the heterozygous. We count all alleles across the individuals constitute the gene pool. We will discuss briefly what gene pool is. For every person, two alleles, two alleles is received, one from each parents. So one is received from the mother and one is received from the father. So let us discuss the key terms before the discussing the Hardy-Weinberg principle. First, discuss what a gene pool is. Gene pool is the total collection of the all genes and the different forms of alleles present in the population. It, is, it represents the overall genetic diversity of the populations. Suppose we consider gene with two alleles, A and capital A and small a. The gene pool includes total number of AA alleles contributed by individuals. The larger or more diverse the gene pool is, the greater potential for variation and adaptation in future generations. Uh, suppose it's a chromosome and it's another chromosome. So it's another. So allele is present in these chromosomes. So combined all these alleles is known as the gene pool in a population. So diverse is the gene pool. There is a because there is a potential of variation because of mutation in this gene pool and adaptation in the future generation. The genotype is the genetic constitution of an organism of a trait represented by pair of alleles it carries. Whether it is capital A or capital small a, it is these are homozygous. These both are the homozygous and it's the heterozygous. The phenotype is observable what is the difference between the phenotype and genotype? Genotype is the genetic constitution of an organism, but phenotype is the observable characteristic of a trait and organism, such as height, color, behavior, which we can observe directly, and these are produced by the interaction with the genotype and environment. Mutation is a sudden heritable changes in the DNA that creates new alleles. Although rare, the mutations are the source of genetic variation, and it's a raw material for the evolution, because the mutation is to the different phenotypes, and phenotypes which goes through the natural selection, and the fittest phenotype will give led to the evolution. That is a majorly migrating population. So after a few generations, these blue ones population, this blue population shifted and inhabited in a new place. So they reproduce among themselves, and after a few generations, we found that there are only blue alleles. So uh, here the gene pool is looking quite different, but from here a small population transfer here, in which red is predominant, and from here the another population transferred after a couple of generations, in which blue allele is predominant, and they reproduce among themselves to give a population where a blue allele, only blue allele is present. So it's genetic diversity of this population. The genetic diversity of this population is quite different from the genetic diversity of this population. So this is a founder effect, and this occurred purely by chance. So it is a kind of a genetic drift. So this is one type of genetic drift which occurred due to the founder effect. Another type of genetic drift is a bottleneck effect. In bottleneck effect arises when a large population undergoes a drastic reduction due to the natural events like disaster, epidemics, or human activity, and the surviving individuals carry only a limited portion of original genetic diversity. Of course, the natural disaster occur purely due to the chance, and the genes that remain uh, only purely due to the chance, not due to their natural selection or survival, which gives them survival or reproductive advantage. It is occurred purely due to the chance, because natural disaster due to a chance accidentally favored one type of allele. So even if population later increases in size, the genetic variance remains reduced, making it vulnerable to further change. Suppose we see it's a population, then what happens in the bottleneck effect? So why it's called bottleneck? Because the alleles remain in the bottleneck, get separated, and surviving in the hazardous event like a natural disaster or epidemic. So so it remains as a survival population. This remains as a survival population, and this new population, there is a more of red alleles in this population. There is a mixture of red, blue, and green alleles. We can denoted different alleles denoted by these colors, but uh, when these alleles are separated, then we can say that there are alleles denoted by red is more in number than the yellow. The alleles denoted by yellow are very quite less here. So the genetic diversity of this gene pool looks quite different from the genetic diversity of this gene pool. Natural selection is a process in which heritable variations that improve survival and reproductive success are favored. Because they improve survival and reproductive success, these alleles transmit from the generation to generation, whereas the other alleles reduced in frequency. This causes changes in the allele frequency and causes new phenotypes, because mutation is the driving force of evolution, and when mutation occurs, these mutations might be favored through the natural selection and may even result in the speciation, because mutated alleles may be favored by the natural selection. Individuals possessing such advantageous traits produce more and have a greater number of progeny, because it provides the reproductive success and improves survival. So natural selection can be of three types. One is a stabilization, in which population acquire mean character value. Suppose there are three types of worms. Three types of worms is there. One is a red worm, one is a yellow worm, and one is a red worm with yellow spots in it. Okay. Suppose so this population, this middle population, carries out the average rate of both uh, this population and this population, and this is the average of these two population. So if this population is favored, this type of natural selection we call stabilization. In this type of natural selection, more individuals acquire mean character. This reduces variation around the average, which is in between these two. This is the average, and maintains the population in a stable form over time. So extremes are reduced in these populations and average is favored. So this type of natural selection is called stabilization. There can be another form of the natural selection where this red population can be favored, or yellow population can be favored. This where extremes are favored. So this is a directional change. More individuals acquire value other than the mean character value. This shifts entire population towards one extreme of the trait distribution. And another type of natural selection is disruption. So this in this type of population, the average population get diminished, and both this population will be favored. Both these extremes are favored. So acquire the peripheral character at both ends of the distribution curve. So these extremes are favored. This reduces the number of intermediates and increases the number of extremes, sometimes leading to formation of distinct groups. So let us discuss about the Hardy-Weinberg principle. In Hardy-Weinberg states that in a large randomly mating population, allele frequency remain constant from generation to generation in absence of evolutionary forces. So evolutionary forces need to be absent for Hardy-Weinberg equilibrium to be true. This condition is called as the genetic equilibrium. Now let us come to the principle itself. Suppose a gene has two alleles, capital A and small a. Let the frequency of capital A is P, and the frequency of small a is Q. So there are only two alleles, and the sum of their frequency must be equal to 1, uh, according to the Hardy-Weinberg principle. So P plus Q is equal to 1. Now in a diploid organism, the possibility of genotype AA will be P square, because there are two P multiplied as if the probability of one allele is P, and the expected genotype frequency of allele is A, then the expected genotype frequency will be P square according to the law of probability. And the probability of small a will be Q square, and the probability of heterozygous, which occurs in 1 is to 2 is to 1 ratio, because we know 1 is to 2 is to 1 ratio it occurs. So the heterozygous frequency will be 2pq. So we get p square plus 2pq plus q square is equal to 1. So this is a binomial expansion of p plus q square. This can be explained from the Punnett square representation. So this is the probability p square, and this pq, these two pq's are added to get 2pq, and this is the q square. So if the p p gets decreased, then the q will increase. If p is 1, then that point q is 0. If p is 0.5, q is 0.5, these two curves. So how p plus q, p and q represents, and this is 2pq. When so it is 2pq frequency is maximum in the middle. So heterozygous will be of maximum frequency. Uh, so this curve shows how the allele frequency behaves. So Hardy-Weinberg principle is important. Why? Because it provides a baseline model for studying evolution. The population it observes genotype frequency differ from the expected value. indicates evolutionary forces are acting, because earlier we told that if the condition that evolution should not be acting for Hardy and Weinberg principle to be true. For principle to hold true, the following condition must be satisfied. There should be a large population size, because large population size there will be a no genetic drift, because if population size will be small and the genetic drift will occur, then allele frequency get disturbed easily. So P and Q get disturbed easily, and P plus Q want to be 1 in that case. So there should be a random mating, there should not be a preference, so that homozygous heterozygous frequency remains uh, constant, as we saw in the Punnett square. There should be no mutation, because mutation creates new allele. We are talking about two alleles here. If a mutation occurs and a allele of frequency R occurs, then it will have some frequency, suppose 0.2. Then in that case, P plus Q will be 0.8, and Hardy-Weinberg can be true in that case also. There should not be no migration, because population, because migration can lead to the founder effect or bottleneck effect, and this will also disturb the allele frequencies. In that case, P plus Q want to be 1, so in that case also Hardy-Weinberg equilibrium can't be true. So this is the case of Hardy-Weinberg also. No natural selection, because natural selection will give rise to the preference to a particular allele. So if it favors the allele P, the frequency of P will increase, and it may give rise to the new alleles. So the P plus Q will be disturbed. So it also is not favored and Hardy-Weinberg equilibrium. Let us go to the questions. Few questions for solving Hardy-Weinberg equilibrium. Is asking the frequency of the population A is 0.62. What is the frequency of allele A? As we know, P plus Q is equal to 1. We have already given allele frequency A with the frequency P 0.62. So frequency of allele A will be Q. Q will be 1 minus P. As we know, P plus Q is equal to 1. So 1 minus 0.62, which is 0.38. Number of alleles. What is the number of alleles? So number of alleles, number of A alleles will be, the population has 5 diploid individuals. So there will be 1000 alleles, because one person can take one diploid individual called 2 alleles. So if the frequency of population allele A is 0.62, then there are 1000 alleles. We can find out the multiplied by the frequency, which gives us to 620 alleles, allele A. And how many A alleles will be required? As we know, we can find out with the Q alleles. The frequency of Q, we know 0.38. We can multiply it by total number of alleles and find out the Q alleles. So A alleles will be 620, and small a alleles will be 380 with Q frequency, small Q frequency. There is another question. In a population, the recessive phenotype occurs at 9%. Assume the trait is controlled by a single locus with allele A and small a. Find the PQ. So we know recessive phenotype is 9%. So Q square is 9%, because Q square is the recessive phenotype frequency, as we studied earlier. So the Q will be 0.3. From the key, we find out the P is the frequency of dominant allele, so it will be 0.7. So genotype frequency, so the genotype, so dominant phenotype frequency will be 0.7 square, as it was studied earlier, 0.49, and the frequency of dominant homozygous will be 0.49. The frequency of dominant heterozygous will be 2pq, which is 2 into 0.7 into 0.3, the frequency of two alleles, and the recessive phenotype frequency will be 0.9. We can, if we add all this, we can still find out that it is 1. The population has 2000 individuals. So how many of each genotype expected? Because dominant homozygous dominant are 0.49 frequency, and the individuals are 2000. We can expect 980 individuals of dominant homozygous frequency. Of dominant heterozygous frequency, we can expect 0.42 by multiple 0.42 into 2000, which is 840, and recessive phenotype can be 180. So these are the frequency. So today we study Hardy Weinberg principle and the problem, how to solve problems, mathematical problems associated with it. Thank you.